Hi everyone, my name is Sabine Wintergerst. I'm the seed lab manager at Fairchild Tropical Botanic Garden.
Hi, and I'm Lydia Cuni, the field biologist.
We are really excited to talk about our ongoing project with four Asclepias species that are locally rare here in South Florida. And we're really grateful because this work was actually funded by the Florida Native Plant Society. And the grant had the title Optimizing Cultivation and Increasing the Availability of four locally rare Asclepias species, an important wildlife supporting plant. And I have to emphasize that this is still work in progress, but we're really excited to show you what we have done so far.
So some background about the Asclepias or milkweeds that you find in Miami-Dade. In Miami-Dade County, we have a limestone rock ridge, and this is a foundation of a lot of the different ecosystems. Here we have the pine rockland. We also have some marl prairie and the ecotone in between. And as you might very well know, much of this habitat has been converted. We have very little natural areas left. And so the 8 native milkweeds, Asclepias species that we had in this area, at least 2 are historically known, and the rest are either listed as critically imperiled, or rare in South Florida by the Institute of Regional Conservation. Some more background, the milkweeds are abundant nectar sources for insects, and they're also in high demand for butterfly gardeners. And there's really no offerings of native milkweed plants in nurseries. So the only milkweed that you can find is the non-native tropical milkweed. So we propose to work towards the conservation of 4 native, rare, or imperiled Asclepias species. The 4 species of our project are Asclepias lanceolata, Asclepias tuberosa, Asclepias verticillata, and Asclepias viridis. So the first project species, the world milkweed Asclepias verticillata, has a IRC designation as rare. It's been recorded for 6 Miami-Dade County owned sites, but I'd like to mention that some of these populations, at least 2, haven't been observed in the last 2 decades. So they're really rare plants here in Miami-Dade County. The leaves are very fine. They're very narrow and linear. They're opposite or can be whorled. The flowers are small. They're white and cream colored, and the fruits may be light green, but here in the photo, we have some light pink and purple fruit pods, which are called follicles. The next species is the green antelope horn, Asclepias viridis. And here in Miami-Dade County, it's just found on the rock ridge. It has a designated status of imperiled. And I want you guys to look at the map on the bottom left. Here in Miami-Dade County, this is a very disjunct population. So the rest of the places where you can find this species is more North and Central Florida. So it's really disjunct and separated from the rest of the state. The butterfly weed is here in Miami-Dade County, Asclepias tuberosa subspecies Rolfsii. It's been recorded for 27 sites. The leaves are very different than the other subspecies of tuberosa. So they're thick and they have wavy margins and very short leaves. And the butterfly weed has many flowers in one cluster. So more than 15 in one cluster. The next species, the 4th species is the few-flowered milkweed, Asclepias lanceolata. And this has an IRC designation as rare, and it's just recorded for 2 Miami-Dade County owned sites and 1 of the South Florida Water Management District. Now, you can also find it in Everglades National Park, but there's no permissions to collect there, so that is another site. And this clusters of flowers are few flowered, so there's just can be 5 flowers in a cluster, not as many as the butterfly weed. So the objectives for our project were to, one, collect the seeds, and number two, once we had the seeds, we would learn how to optimize, how to germinate the seeds. We were also interested in testing the long-term cold storage viability of the seeds. And once we had some seeds germinated and we generated plants, we were interested in studying number 4, optimizing the horticultural protocols. Number 5 was to then create a living ex situ collection of all these 4 species. And then once we had an ex situ collection, we were interested in distributing some of these plants. So objective 1, to collect our seeds, first we had to determine where these plants could be found. So we created a matrix of the species and the different sites. And that gave us some direction of how to proceed, whether we needed to visit an herbarium to find some historic information. And also we have a lot of records here at our conservation program. So we did some data mining of our records, and that all gave us some direction of where we can find these plants. Some are very rare here in Miami-Dade County. And to collect those seeds, once we did our surveys and we found fruiting plants, we used drawstring organza bags that we would place over the seed pod so our seeds would not fly away as a milkweed plants have seeds that do. And we would bag the pod and revisit to come back and collect our seeds. And I would like to note that during this project last year, there was some prescribed fires at multiple sites. And these prescribed fires had good results for the Asclepias species. So there were many resprouting and flowering and some pollination. And we rediscovered some populations of different species. And we were able to get a more accurate count of how many are really out there at some of these sites. So some results from the seed collection. So for Asclepias tuberosa, we've been able to collect 600 seeds from 7 sites from 10 different plants. For Asclepias viridis, we've collected 400 seeds from 5 sites from 5 different plants. And for the world milkweed, Asclepias verticillata, we have collected 76 seeds from just 1 site. It's very rare, and from 5 maternal lines.
And so once we had collected some seeds, we actually started to work on optimizing the seed germination. And for that, we tried a few different seed treatments, and we also sowed the seeds in different media. And here on this slide, you can see the results for the Asclepias tuberosa seed germination. So in that case, we sowed seeds from different locations and recorded how the percentage of germination after, depending on the day since sowing. And so the different colors represent different populations or the seeds collected from different sites, and the numbers next to the line indicate the number of seeds we actually sowed. And the numbers are different because it depended on how many seeds we actually had available from that site at that time. And you can see without any pre-treatment, we got a really good germination rate of 80% and above. The only seeds that didn't germinate that well were from the Pine Shore Pineland Preserve here in yellow. And that might have been just the seed quality, because sometimes the seeds we collected in the wild, you could already see that the quality is not as good as it could be because the seeds look crinkled and a little small. And so that was probably the reason for the low germination rate here. What I did want to mention before also for the Asclepias tuberosa, it was interesting to see that we didn't need any seed pre-treatment for this species, because normally when you look to grow tuberosa in different parts of the US, it's normally recommended that the seeds need cold stratification to germinate. But in our case, we didn't need that, which makes kind of sense because here in South Florida it doesn't get that cold. But it was interesting to see that it really didn't need any cold stratification to get a high germination rate. And then for here are the results for Asclepias viridis. And here actually we found that if the seeds were not pre-treated at all, which is the orange line, we had a relatively low and slow germination rate. So it leveled off at around 40%, and it took a really long time, like more than a month to get there. But when the seeds were desiccated, which was the case for the blue and the dark green line, the seeds germinated much faster and the germination rate reached up to 100%. And here in the photo, you see another problem that we encountered with the Asclepias viridis seedlings is that they seem to be very tasty to little iguanas. And so we actually had to cover the pots with a little cage to protect them, because here in the picture, you can see how a lot of the seedlings already got eaten right away. And here are the results for the Asclepias verticillata. We also tried to desiccate them, or to desiccate or not desiccate them. And here you can see that it didn't make any difference, and both treatments led to really high germination rate of 100%. And maybe you noticed in the pictures also the different media we sowed the seeds in. So we used either standard potting soil like here, or the other pictures, we sowed them in turfis, which is a clay product, which we had some success with other pine rockland plants, but in Asclepias, it didn't seem to make any difference in which media we sowed them. So we stuck with sowing them in standard potting soil. And so one other thing that we wanted to try, because of Lydia's observation that a lot of these plants seem to grow after fire, we wanted to see if the seeds also react to a treatment with smoke solution. We had some success with other pine rockland plants that produce seeds that are really reacting to the exposure of smoke, and that the smoke actually triggers the seed germination. And so we wanted to try this with our Asclepias as well. And this slide just shows you quickly how we produce our smoke solution. So we use a bee smoker, and we put pine straw in it and let it burn there. And then the smoke goes through the hose into a sidearm flask that is filled with water. And the hose actually is submerged in the water. And so the smoke has to go through the water and then get sucked out with this tube. And that leads to all the water soluble chemicals of the smoke that are now trapped in the water and can be then basically used to treat the seeds. So you end up with this bottle of very strong smoke solution that we keep in our freezer and use whenever we need it. And here you can see the results of this effect of smoke solution on the seed germination of the 3 species we had seeds from. So on the left and in blue, you see the results for Asclepias verticillata, and dashed line is the control, which means these seeds had no treatment with the smoke solution. And you can see that in this experiment, the germination rate of the Asclepias verticillata was generally not that good. But that was because at that time we didn't have that many seeds left, and they also were a little crinkled and didn't look as good as the seeds we had before. But when you look at the smoke solution, you see that the germination rate is even a little lower than it was before. So it definitely doesn't help in this case. And then the results for Asclepias tuberosa in orange. Again, the dashed line is the control, and the solid line is the results for the seeds that were treated with smoke. And here you can see that it didn't really make a difference. So they germinate both really well, and the smoke solution didn't change that much. But in the Asclepias viridis, you can actually see that the seeds that were treated with smoke, which is the solid line, they actually seem to germinate a little faster with the smoke than without the smoke. But at the end, they reached the same germination rate. And yeah, we only tried it once, and we didn't include it in our usual protocol of growing them. Another objective was to study the long-term cold storage viability of seeds. And even though at the beginning of the project, we already knew that Asclepias generally, they survive being frozen in for at least a short period of time, but we don't know how long we can keep them in our freezer. So here on the right, you see a picture of how the seeds are actually sealed in these silver mylar bags to protect them from humidity. And that's how we store them in our freezer at minus 18 degrees Celsius. And we started a germination at a storage trial by freezing a bunch of seeds. And we're going to test every 6 months the viability of a subset of these seeds. And so here you can see our first results from Asclepias viridis. On the left, you see that the termination of the fresh seeds in the blue line, and then the termination of the seeds that were frozen for 6 months, which is the orange line. And you can see that they might even terminate a little faster, but definitely the termination rate still stays the same, which is good news. And the same is true for Asclepias tuberosa. You also see the fresh seeds in the blue line, and the orange line is the seeds that were frozen for 6 months. And this trial, the termination testing is actually still ongoing, but it looks like it's going to be a very similar termination rate. So the next step was to actually try some different growing methods for the plants. And one of the first things we tried are different pots. We either use these deep black deep pots, which were we already predicted that that might be a good pot to grow these Asclepias because they normally have really long tap roots that really want to grow down into the ground. But we also tested these 3-inch pots because at the end, our goal is to distribute the plants. And we just found that distributing plants in these tubes is a little more challenging because they don't stand on their own, and people just really like more the 3-inch pots to take home with. And so we wanted to see if it's also possible to grow them in the 3-inch pots. And on the left, you see how the seedlings survived after 5 months in the different pots. So in green, you see the 3-inch pots, and in blue, it's the deep black pots. And you can see that for Asclepias viridis, it didn't make any difference. They both survived really well. But the Asclepias tuberosa seems to really grow better in the deep pots because when we grew them in the 3-inch pots, almost 50% of them died. The Asclepias verticillata, on the other hand, they seem to be fine in both pots. And here in this picture, I just wanted to show you how different the roots look between the two pots. So in the upper row, you have the 3-inch pots, and in the lower row, the deep pots. And you can see, for example, here in the Asclepias viridis, you can really nicely see how the thick taproot actually gets bended and grows to the side in the 3-inch pots. And also here in the verticillata, you can see how the roots they try to grow further down and it kind of gets stuck here. Whereas when you look at the roots of the deep pots, you can see how they nicely grow all the way down, especially here in the verticillata, you can see it really nicely. Another thing we tried was different growing media. So we grow plants in we call it the rocky mix, which is 1/3 of standard potting soil, 1/3 of crushed limestone, and 1/3 of turfis again. The turfis is these brown little bits in the mix. So it's basically chipped clay, and it makes the mix really, it doesn't retain that much water, so it's draining really well. And the other mix we used is a sandy mix, which is also 1/3 of standard potting soil, 1/3 of crushed limestone, and 1/3 of silica sand, which does retain water a little more than the rocky mix. And when we look at the survival of Asclepias seedlings after 4 months, we found that the difference is definitely not big, but we found that in Asclepias viridis and Asclepias tuberosa, that they survived a little bit better in the rocky mix, which is the orange bar. So we decided after that we just keep growing all the other plants in our rocky mix. And now I just wanted to highlight some of the more problematic things we encountered of growing the Asclepias. So one was that especially the Asclepias viridis, but the other 2 species as well, they're very attractive to aphids. And so here on the left, you can see a picture, and this is a very typical picture of a plant that we had in our nursery. And so we started to treat the plants with neem oil every 2 to 3 weeks, and that helped to keep the aphids at bay a little bit. And then the other problem that we encountered in our Asclepias production is the transitioning from the greenhouse to the outside growing area. So normally we sow the seeds in a community pot and then repot the little seedlings into their individual pot after that. And so we put them in our greenhouse so that they can get acclimated to their new environment. And that works really well. And almost 100% of them survive that step. But the more tricky part is moving them from the greenhouse actually to the outside. So we already put them in a shady place so that they're not exposed to full sun right away. But what we encounter is that most of the seedlings turn brown and lose all the leaves once they're outside. And luckily, most of them do resprout, and after that, they're fine. But that's the step where we lose around 30% of our seedlings, and even trying different watering regimes or different length in the greenhouses, so far we couldn't really avoid this loss. And another thing we noticed during our production of Asclepias tuberosa is that Asclepias tuberosa really flowers several times a year in our nursery. But so far, we also sadly didn't have any pollination, and they never developed any seeds. And so what we found is that they flower, but then once the flowering is over, the plant actually starts to die back. The leaves fall off, and it looks really bad. The first time we were really afraid that all of them are going to die. But what they do is they die back, and then after a few weeks, they start to resprout, and the whole cycle begins again. And so with all these things we learned, we were able to grow around 200 Asclepias plants at our nursery so far, 83 tuberosa, 43 verticillata, and 74 viridis from several maternal lines and several populations. And yeah, and the only species that actually produces seeds so far is the Asclepias verticillata. So here you can see it flowering. And we did actually observe a scarlet wasp regularly on the flowers. So that's probably the pollinator. And by now, just this week, almost 90% of our Asclepias verticillata plants are producing seeds, and we start to collect them, and already collected like around 300 seeds. So we're going to have a lot of seeds from this species, which is really great. And for the other two species, we actually did try to hand pollinate them. Here you can see Lydia trying it on Asclepias viridis, and me too. But so far, sadly, we didn't have any success with that. I mean, we hope or think that's just an issue of practicing because it is really hard. So we hope this growing season will get some seeds from these two species as well.
So we generated some of those 200 plants, and the next goal was to start distributing these plants. So our first step was to plant some at the Pine Rockland plot at Fairchild Garden. So we planted a total of 18. 4 of those were Asclepias viridis, 8 were Asclepias tuberosa, and 6 were Asclepias verticillata. And from those, we think just 1 Asclepias tuberosa might not have made it. Something we experienced after the outplanting, there was some iguanas at the garden that found these plants tasty. So we had to implement these cages that you see on the left to protect the plants from any further herbivory and to give them a chance to regrow. All the species were kind of affected, but somehow this Asclepias viridis was left alone. Maybe it was too tall. They couldn't eat it all. So we have one there that's blooming right now. Another thing we experienced during growing these plants, we found those monarch and queen caterpillars that everyone gets these plants for, for butterfly gardening. So we had the monarch caterpillars in the nursery last year in August 2020. And this has also been observed in the wild. Here on the bottom right is in the wild a monarch caterpillar on the Asclepias viridis in April. So, and also when we were doing these surveys, we found in Asclepias a queen caterpillar on the Asclepias verticillata. So looking forward, we're excited to continue to try to have seeds produced ex situ, and we're going to continue doing some seed collections in the wild. We still need to collect seeds of the Asclepias lanceolata, and we're going to still visit some new sites that we haven't visited yet, and also try to collect seeds from new genetic individuals at some sites that we've already visited. And we also are going to begin distributing more plants to some foster gardeners in our Connect to Protect network. And thank you.
Thank you everyone for listening to our talk. And we also want to thank especially the Florida Native Plant Society and Conservation Committee for funding this work and the support from the state chapter that we got for that. And all the other people that were involved in helping with seed collection or growing the plants. And also, of course, all the Miami-Dade County parks and lands and natural areas management for providing us the permission to collect the seeds.